Okay,
good.
thanks, Tom.
Welcome, everybody. Thanks a lot for coming today to Joe Oshesky's Master's Exit Seminar. Before I start my announcements, I would say, if you're in town and interested, I'm having a social at my house at 5 o'clock today. So you're all invited, especially if you know Joe. I live at 734 South Lafayette, 5 o'clock, so that it'll be started and done quickly. And I'll have drinks and snacks for you there if you want to come. Okay, just congratulate Joe, we hope. Joe is originally from Rochester, Illinois, in central Illinois. He was a transfer student to Western. He got an associate's degree from Lincoln Land Community College. I met Joe in genetics class that he took with me. It was post-COVID that first year, so he was masked. So I actually didn't see him for 2 or 3 years after that. But it was clear, even though he was masked, that he was a super hard worker. I think worked obsessively to be at the top of his classes. That's true. Okay, so while he was a student, in addition to studying hard, he worked for WEAMS, the Western Emergency Medical Service, and he just recently finished a training program and is now a certified EMT, Emergency Medical Tech. For 6 years, he has been in the Air National Guard. He's currently a staff sergeant. So for his work today, I want to say it's been really fun for me to work with Joe. It's been a true collaboration. This project was originally my idea, but he took it and ran with it. He collected more data than I had planned. He did statistical analyses that I didn't think of. It's been fun, but I've had to learn a lot to keep up with what's been going on here today. So I told him I would keep this brief because he told me he wants to talk a lot. And so Joe's title is Where to Look, Tissue Tropism of 5 Protozoan Parasites and 1 Bacterial Pathogen in Peromyscus leucopus in Western Illinois. So, Joe.
All right, sweet.
Thank you for the introduction. I want to preface by saying 2 things. One, I want to say some scientific names. I apologize if I say them technically incorrect. I'm still learning. And 2, I want to thank everybody here in person as well as on Zoom for showing up. It means a lot to me. I've spent a lot of time working on this project. As Dr. Meager said, the title of my presentation is as follows. With that, if I was to give a quick project preview, you may not know, but here in the United States, the number of vector-borne diseases has more than doubled in the past 15 years, right? With an ever-growing increase in the total number of these diseases, little is known about where they are located, where at geographically. Are they here in the United States? Are they here in Illinois? Are they this far in Illinois? A lot is unknown. Specifically, I'm going to be looking for these 6 pathogens, many of which are vector-borne in nature, with the exception of Toxoplasma gondii. With this, and these particular pathogens, you can either check the vectors or the reservoir hosts themselves. In this study, I'm going to look at 60 white-footed mice, which serves as a reservoir host for the majority of these pathogens, with the goal to try to answer the question, how is that?
Distracting me.
Go, please keep going.
Yeah, so with the goal of trying to ask and answer the question, where to look. So a lot of these particular pathogens, once they infect a mouse or another host, they reside in very specific tissue locations. Therefore, I used a combination of PCR along with gel electrophoresis to look at 3 tissue samples per mouse, 1 from the blood, 1 from the spleen, and 1 from the ear, with the idea to see, can these pathogens infect mice? If so, where at? So we take a look, quick introduction so we're all on the same page. What I want you to visualize here on the graph is that as you move down, you get more and more specific. If we take a look at infectious disease, think any illness caused by a pathogenic microorganism. There's many different types. You got bacteria, you got protozoans, viruses, fungi, you name it. With this, it is one of the major causes of death worldwide. So it's really important to study. The issue is, this is such a broad term, right? You simply cannot study every single pathogenic organism at one time. What I did, I focused primarily on what are known as zoonotic diseases. Think first 3 letters spell out zoo, animals. These are infectious diseases transmitted from other animals to us humans. They account for roughly 60% of all human infected diseases, So it's still a pretty broad term, but we're getting a little bit more specific. Vector-borne, right? Think any disease that requires a vector for transmission. Simply put, a vector is anything that can transmit a pathogen from one host to another. Here in the United States, ticks, as seen on the bottom left or bottom right, are the primary vector in which I looked at. In other parts of the world, mosquitoes and fleas serve a critical role. Of this, they account for roughly 17% of all infectious disease, with an estimated death of unfortunately 700,000 people who come into these pathogens each year. It's really important to study, get a better idea of where these are located. We zoom in all the way at the bottom, we focus in on tick-borne disease. Simply put, any disease that requires a tick for transmission. Here in the United States, roughly 75% of all vector-borne diseases are tick-borne in nature. So it's really critical that we get a better understanding of the pathogens that cause these tick-borne diseases. As you can see, there are 3 main tick species of interest because they deal a lot with human infecting diseases. We have the Lone Star tick, the dog tick, and the black-legged tick, also known as the deer tick. For simplicity, I will not say the scientific names, I'll simply say the common names. Well, the entire point of this graph here is to show that these 3 tick species are found throughout the state of Illinois. Now this graph is a little outdated, as you can see the counties in white here, there's simply no data. But we know these ticks are found throughout the state. We zoom out to all of the United States, what you will see is that Illinois is a darker shade of blue, corresponding to a higher number of tick-borne diseases. Therefore, especially here in this state, it's really important we get a better idea of what is here. I'm going to quickly go through each pathogen as to why we should even look for it. First, if you know anything about tick-borne diseases, you've probably heard of this one in particular, Borrelia burgdorferi. Primarily transmitted by the black-legged tick or the deer tick. And if we take a look, many of you may recognize the sign and symptom as it is the major cause of Lyme disease here in the United States. And it is the most common vector and tick-borne disease, therefore accounting for roughly 82% of all tick-borne diseases. So this pathogen in particular is really important to study. A lot of research is focused on this one in particular, but not a lot focused on the other pathogens listed here in a second. These roughly show the life cycle. They're very complex, right? And once we get onto the protozoans, they're going to be even more complex. But all I want you to do here is focus in on the animals associated with each one of the pathogens. So as you see, there are different types of mice or small mammals. Second one is Babesia species, primarily found inside of red blood cells, so very, very small. Again, primarily transmitted by the black-legged tick. And there were a couple species I was interested in potentially finding. We got Babesia microti, Duncani, as well as Divergent. If you notice, Babesia microti is bolded because this causes human babesiosis and is the most common blood transfused parasite here in the United States. So if it's here in Illinois, we need to know about it so that we can prevent blood transfusions of this particular parasite. Notice, very complex life cycle involving multiple hosts in which multiple life stages. But note the mouse indicated here. Cytoxoan felis, which is a protozoan tick-borne disease, is also found primarily inside of red blood cells with the addition of different types of cells, and is primarily transmitted by the Lone Star tick, but there is some evidence showing that the dog tick can serve as a potential vector. This causes cytoxoonosis, and for you cat lovers out there, this is very important because it causes a potentially life-threatening disease in domesticated cats. So if your cat was to be bitten by one of these 2 tick species that's infected with this protozoan, potentially your cat may die. Even with proper treatment, roughly a third will succumb to this pathogen. It's really nasty. Take a look, complex life cycle, multiple life stages, but there is not a mouse listed here. That is key. That is really, really important. It's mainly associated with bobcats as well as domesticated cats. The Hepatozoan. Notice in the top right, it says protozoan tick consumption. So this, instead of a tick biting and passing along this pathogen, instead what happens is some animal will consume a tick that's infected and become infected itself. Some species I was really interested in seeing if they are here are Hepatozoan canis, as well as Hepatozoon americanum. Due to the fact that they cause hepatozoonosis, which is one of the most significant parasitic diseases in dogs. I personally love dogs. I do not want my dog to come into this particular pathogen. Take a look, complex life cycle, note mouse is listed in the top left. Trypanosoma species. This is a vector-borne disease, not a tick-borne, in the sense that it's transmitted primarily by what are known as kissing bugs. These are about the size of your thumb, and they tend to target the area around your mouth and give you a kiss. So it's kind of disturbing. I do not like how these look. They look really intimidating. But of interest, I was really curious as to whether or not I could find Trypanosoma cruzi as well as Trypanosoma peromyscus. The bolded one, Trypanosoma cruzi, because it causes Chagas disease. Those who are unaware, this affects roughly 7 million people, of which a small proportion dies, but those who live face really debilitating signs and symptoms. It really messes with your quality of life. If we take a look at its geographical distribution, it's primarily located in South America and Central America, but if you notice, it's starting to creep north. All right, one of my committee members, Dr. Zeman, has found this in the state of Illinois, and I'm curious, is it this far north? Additionally, when I was doing some background research, I found this study published just last year in which it is showing the counties of Missouri and Illinois. Anywhere you see a dark or black region, think they found one of the vectors capable of passing along this disease. This is pretty big. If the vector's here and I can find the pathogen, humans are definitely here, right? So potentially we may see some cases of Chagas disease here in Illinois. Complex life cycle, note the mouse down in the bottom right. Last one is Toxoplasma gondii. Multiple different life stages, really small. These are inside of cells, and it causes toxoplasmosis, which is estimated 40 million people here in the United States are infected with this disease. And unfortunately, lifetime infections can occur. If you're pregnant or planning to be pregnant, please be aware of this particular pathogen, because potentially it could result in a miscarriage and or serious developmental issues for your developing infant. This is the one in which if you visit someplace and they tell you to stay away from a cat's litter box if you're pregnant, this is the disease that they're talking about. There's multiple different ways for humans to become infected, but note the mouse listed here. As stated, you have multiple detection options, right? You can test the vectors themselves. I could go out in nature, catch these ticks, look for the pathogens of interest. Or what I did is you can test the reservoir hosts themselves. Note the mouse. Simply put, think reservoir is any animal that harbors or sustains a pathogen. It's the source or the bank of infection out in the environment. The white-footed mouse, listed here, is a critical reservoir host for many of the pathogens I'm looking for, primarily due to 4 main reasons. One, it plays a critical role in tick feeding dynamics. What do I mean? The majority of these pathogens cannot be what is known as vertically transmitted, meaning the adult tick cannot pass this pathogen on to their offspring, which means that the developing ticks are uninfected when they hatch from the egg. They acquire these particular pathogens by feeding on mice and or small mammals, as seen in the larva stage as well as the nymph stage. Additionally, they're immunologically tolerant, meaning I could check one animal and potentially find all of these pathogens I'm looking for. Very widespread. If we take a look at its geographical distribution across the United States, it's one of the most common mammals. And to compound with that, as humans begin to fragment the habitat, we're going to come into closer proximity to these white-footed mice, all of which make the white-footed mouse an excellent reservoir host to check. I had 4 main research questions with this presentation. 1, do these occur in white-footed mice? And if so, specifically here in Western Illinois. 2, what is or what are the most important determinants of pathogen detection? Do these detection rates vary across the blood, spleen, and ear tissue? As well as do these pathogens display localized or disseminated infections, which I will define later. Quick methods. My research location was in Hancock County, as noted by this red dot, at the Alice Live Kitty Science Station. This is a picture of me with the sign. Really like this place. I've done a lot of work out there, and it's awesome that Western has this. At Kibbe specifically, all of these mice that I'm going to be referring to were caught on 15 long-term plots that have a lot of variation. I did not specifically include and or consider vegetation type or prescribed burn frequency or abundance due to my inability to say a mouse caught at one plot did not happen to go interact with a mouse caught at another. Because this is in a roughly 2-acre area patch of woods and mice are known to move around a lot. We take a look, mice were caught using what is known as a Sherman trap here, and then mice caught would be taken back to the lab and dissected. I was very fortunate in that a previous grad student of Dr. Meagher's let me use her blood and spleen tissue samples that were previously extracted in the fall of 2021. So these mice I'm looking at, I did not catch, but previous students did. And I will thank her at the end of this presentation. Well, that leaves the ear tissue, right? In the summer of 2024, when I was helping Dr. Meagher dissect mice, as seen here, this mouse is dead. It was humanely euthanized. But one of the steps we had to do were check for ticks. And what Dr. Meagher has noticed throughout his extended period of time of looking at these particular animals, that you would commonly find ticks along the ear, as seen here. If I'm looking for tick-borne diseases and they attack a mouse in the ear, why not check the ear, right? So did a little bit of background research, found a couple articles that said potentially I should check this tissue. Talked to Dr. Meagher, luckily he let me do it, and it's really, really fascinating what I found. All these mice were frozen, on which what I had to do was collect ear tissue samples as seen by hole A and hole B, or here. I could not check the entire ear, so I tried to check the distal end or the farthest away, or the proximal end near the mouse to try to get a representation of the entire ear. I extracted the DNA from these, and then I'm going to talk about how you go from a DNA sample to a pathogen detection, right? That seems like a really big jump. How do you do that? First, I use a molecular technique known as polymerase chain reaction, in which I will selectively amplify pathogen DNA. I will then use a combination of gel electrophoresis as well as a positive control in order to compare and determine if my tissue samples are positive or negative for that pathogen. I will then determine the identity and whether or not I truly found that pathogen using DNA sequencing. We quickly go through polymerase chain reaction. For those who are unaware, you use what is known as a thermal cycler, in which what I want you to picture with this image here is this entire gray region. Think that is all the DNA found in one tissue sample. But I'm only interested in this tiny red region corresponding to the target sequence, or think the pathogen's DNA. So if I know the pathogen I'm looking for and the sequence of that pathogen, I can then take advantage of the nature of DNA, run it through a thermal cycler in which it will go through stages of denature, anneal, and extend. And anneal specifically, these blue regions are primers. With this, as a result, as this machine will continue to run, I will get a mass amplification of the specific target sequence I'm after. I will then take all those samples, load them onto a gel as seen in the middle in the right image. From here, what you do is you run a current through this gel. DNA is negatively charged. This gel has a lot of pores through it in which I can separate DNA based off of their overall size. From here, I can use a special machine. Thank you, Eastern, for having this machine in which I can produce an image of the gel. From here, remember how I said I compared it to a positive control or a reference sample? That can be seen in well 1. What you do to determine presence absence is you simply go straight across. I know this is the particular sequence I'm looking for, so I can compare and see, oh, well 3 has that band, well 4 does not, right? Presence, absence, or positive, negative. From here, I could sample and or sequence roughly 48 samples. Since I'm looking for 6 pathogens, my goal was to do 8 positive samples per pathogen. And as noted here, I tried my best to get an equal representation of the blood and spleen, as at the time of sending these samples off, I did not look at the ear. Results, and this is the really cool part of the stuff. So now that we've gone through all the background, now we can answer these research questions. The really cool thing is I can answer all 3 of these, the first 3 at once, using what I'm going to refer to as a multiple pathogen model. Thank you, Dr. Nepal, for bringing up this idea. I looked at all mice across all pathogens, all detection statuses, which seems like a lot, right? And the part I really struggled with was trying to determine what variables I need to consider. Right? If I'm looking for a response variable being positive detection, it's either there or it's not. You have multiple different predictors that can potentially explain and or produce a positive result. What I did was I analyzed all of these predictors, including all possible interaction terms, which I'll describe here in a second, using a GLMM, which just stands for a generalized linear mixed effects model. Mixed effects, I needed to consider that from each mouse, I took 3 tissue samples per mouse. So I cannot say that those 3 tissue samples are independent or did not interact with each other in one way or another. And what I found by ranking all these models using their AICC values is that this was the best model in determining or explaining a positive detection. The mass of the mouse was important, the tissue type, pathogen type, as well as this interaction term, which simply put, the impact of tissue type on the response variable depends on the pathogen type in which you're looking. The AIC value, essentially, the simplest way I can describe it is you are looking for a model that is very simple, meaning few variables, but explains a lot of the variation. So this model just picks what is known as the best overall model. From here, if we take a look, these were the potential variables that were important, but I wanted to determine, are they significant? Are they statistically significant? And what I found were that these 3 shown in red were. But that leaves the question, what is going on with tissue type, right? Why is that included in the best model, but it's not significant? Well, if we take a look with the x-axis representing the tissue type, blood, spleen, and ear, with the y-axis representing a predicted probability, what you notice is that there is very little variation among all the tissue types for a positive detection. Simply put, if you only consider tissue type, there's not a lot of variation. We take a look at mass though, with each panel corresponding to a particular pathogen, x-axis representing the mass, y-axis representing a predicted probability. Regardless of what pathogen you look at, regardless of what tissue type, you will see a slight increase in predicted probability of detection as the mass of the mouse gets larger, meaning larger mice tend to have a higher predicted probability of detection. Very interesting. Additionally, that pathogen type, meaning the easiest way I can describe it is that some pathogens are found more often than others. Some have a higher predicted probability of detection, such as Babesia and Hepatozoan, with other pathogens not having as high of a predicted probability. Well, that leaves this interaction term, right? Simply put, think patterns. If you look at different pathogens, you will notice different patterns in detection rate by tissue type. The easiest way to explain this is if you look at the far left, the left 2 graphs with Borrelia burgdorferi along with Hepatozoan species, you will notice that for Borrelia, if you check the ear tissue as seen in gold here, you have a higher predicted probability of detection compared to the blood and the spleen. But the exact opposite is true for Hepatozoan. Ear tissue is by far the worst tissue to check, and blood and spleen share roughly the same predicted probability of detection. You may also note that some bars are larger, Hepatozoan and Babesia, as explained in that previous slide, they're found more often. So therefore they have a higher predicted probability of detection. And if we look on the far right, these 3 are found in a very low overall quantity. From here, I wanted to determine the relative importance of each one of these variables, right? So I found that they're significant. Well, what amount of variation do each one of them explain? What I did was I ran simulations of my data in which allowed me to calculate partial R-squareds. Simply put, it's the relative impact of each variable while accounting for the other variables. That last part is really big. We take a look at the graph produced. My full model, looking at all of these predictors, explains almost 84% of the variation as seen on the far right, indicated by the full model. If you note, tissue type and pathogen do not explain a lot of the variation, but as you move to mass, that explains a little bit more. But this pathogen interaction term is by far explaining the most amount of variation the entire model can explain. Which simply put, if you are looking for a particular pathogen, you need to consider the tissue type in which you look. But that leaves research objective D unanswered, right? Do they display a localized or disseminated infection? Simply put, think localized. I only found the pathogen in one tissue within a mouse. Disseminated, I found it in 2 or more tissues. What I was very curious in is if a localized infection occurs at a higher proportion than by chance. That by chance is this fraction 3/7, as with 3 tissue types. 3 of them would be considered localized, meaning blood only, spleen only, or ear only, and of which you can make 7 potential combinations. Simply put, I compared this value to the proportion that I observe using this binomial test, and what I found was that the 4 pathogens, Borrelia, Cytoxoan, Toxoplasma, and Trypanosoma, all displayed a localized infection occurring at a higher proportion than by chance. Right? This has huge implications for pathogen detection and research surveys. But this graph simply does not explain where at. Right? It doesn't explain what tissue. It just says primarily found in one tissue. If we take a look at what are known as nested donut plots, what I just showed you corresponds to the inner donut. The exact tissue distribution can be seen in this outer donut here. 3 patterns emerged. 1, for Babesia and Hepatozoan that were found very often, multiple tissues tended to be infected all at the same time. It's very interesting, right? So for those 2 in particular, you may not need to consider the tissue type because you can check, say, if we take a look at this purple region here, you can check the blood or the spleen in order to determine if a mouse is infected with this pathogen or not. The second pattern can be seen with Borrelia and Trypanosoma, in which of the localized infections, they're primarily only found in one very specific tissue type. Right? So for Borrelia, primarily in the ear, Trypanosoma primarily in the blood. Which means that if you were to try to check and or find either of those pathogens, you would likely check the one in which is found most often. So ear for Borrelia, blood for Trypanosoma. The third pattern, though, can be seen on the far right. If you take a look at Cytoxoan and Toxoplasma, they were found at a localized infection, as noted in the inner donut. But if you take a look at the outer donut, notice that that one tissue type in which it was found varies. Varies a lot. If we take a look at Cytoxoan, ear tissue, as well as blood and spleen were found pretty often, right? Which indicates that if you're looking for the far right 2 pathogens, you may want to look at multiple tissues in order to account for the most infection status. That was very, very interesting. Remember those samples I sent off to get sequenced? This is why this part is very important. If we take a look at Borrelia, the thing that causes Lyme disease, right? What I did was I sent these samples off to get sequenced. If you see any of these dashed, simply put, the sample I received back from the company was of such low quality that I could not analyze that particular sequence. But as seen by these bottom 3, it did produce a sufficient sample in which I could compare what I got back to something in a database in which I have listed the best overall match. And if you note, all 3 of them say Borrelia, indicating that I can say with a high level of confidence, I did in fact find Borrelia, or the causative agent of Lyme disease. Cytoxoan, and this is very interesting, right? If we take a look, every sample that produced a sufficient sequence came back the best match, matching with Cytoxoan. This has not been documented in white-footed mice to my knowledge, so this was a pretty significant finding, because every single sample corresponded with the pathogen I was looking for. Same thing can be noted for Hepatozoan, but I do want to note that a lot of the best overall matches, simply put, were Hepatozoan species. Therefore, I did not find Hepatozoan canis or Hepatozoan americana. So those 2 I was interested in, I did not find those. These are likely mainly associated with mice. Trypanosoma. Remember the particular protozoan that causes Chagas disease? Well, some of the samples I sent off to get sequenced, the best overall match was in fact that protozoan, which is pretty alarming, right? The pathogens here, the vectors here, that's not a good combination. But if you note, the other samples I sent off to get sequenced came back as another species of Trypanosoma. So not all of them are representative of the particular pathogen I was looking for, or most interested in. Toxoplasma, I only got one sufficient sample, but it did come back as the exact pathogen I was looking for. So this is why sequencing is very important. So what I've been calling Babesia, despite the fact that my positive control did get amplified, we note it's a Pathozoan. It's not Babesia. Therefore, unfortunately, I cannot say what I'm calling Babesia is in fact Babesia, because it's matching more closely with Hepatozoan species. To conclude, some key findings can be essentially split up into two main groups. One, that pathogen detection depends heavily on the tissue type in which you examine, right? That interaction term was by far the most significant in explaining the overall amount of variation. And some pathogens displayed a localized infection at a higher proportion than by chance. Meaning, to answer that question of where to look, is that important? By far it is. It's very, very significant. Additionally, I found that genetic sequencing is critical for identification, right? I was able to say with a high level of confidence that I found 5 of the pathogens I was looking for, excluding Babesia, unfortunately. Note, I was able to say with a pretty high level of confidence that I found Trypanosoma cruzi here in Hancock County, Illinois. It's pretty alarming. As well as I found, and or all my committee members, additionally, we found Cytoxoan felis and potentially a new non-felid host, which is pretty significant, especially for all you cat lovers. White-footed mice are around, cats are around. Potentially that's not a good combination. Simply put, to conclude, not only is genetic sequencing absolutely vital for PCR detection of pathogens, but you definitely need to consider the tissue type in order to find these pathogens. With that, I want to give some thanks. One, for the funding for this project. School of Graduate Studies, I really appreciate it. Biological Science Department for some scholarships. I also want to thank some previous students for their help. Sarah, I really, really appreciate it. She was the one that let me use her blood and spleen samples. Kevin Delgado did a lot of the primer and background research, as well as Evan London, who ordered the primers. These 3 people essentially laid the foundation on which this project was built. I'm really thankful for them. Also, huge shout out to all the faculty here and all the friends I've met along the way. I've absolutely loved my time here at Western. I really don't want to leave. I love this place. I also want to thank my family, dad, mom, brother, as well as my grandpa for letting me do this project. I spent a lot of time on this project, so I really appreciate all the help you guys did. And big shout out to my committee members, Dr. Hum, who's in the audience. Thank you very much. You made a huge impact on me for my time here at Western. Dr. Zeman, thank you so much for letting me use your lab, for always being open to questions. I really appreciate it. Project would not have been possible without you guys, as well as Dr. Meager. Thank you for giving me this project and for spending countless hours of your life editing my thesis and helping me put together this presentation. So I really appreciate it. I really like this picture. This was my last ever class at Western. Dr. Meager recently retired. This was his last ever class. This was the last slide from the last presentation. That's kind of cool in my opinion. References, and thank you guys for your time. Thanks.
So there time for questions if anyone has one for Joe. Stop sharing. Yeah, it's okay. Okay, are you there? Okay. So I see that you're in charge of fielding.
Sam, what do you got?
Okay.
Hey, Joe. Hey, Joe. A while back, we talked about the tissue by pathogen interaction, how it's located in specific tissues, right? Like the ear.
Yes.
Or was it the liver, the spleen?
Spleen.
The spleen?
Yeah, thick blood, spleen, and ear.
Right.
Yeah.
Have you learned any more, done any more looking into like the mechanism for what keeps pathogens in specific tissues?
As to explain why I found a pathogen in one area versus another.
Yeah, like why doesn't it like go from the ear to the rest of the body?
Yeah, that's an excellent question. And that brings up the idea of tissue tropism. Unfortunately, a lot is still unknown, but some potential explanations that I've thought of are one, especially for tick-borne diseases, maybe I just caught that pathogen at the right time. Maybe that mouse happened to have a tick pass along the pathogen, and I was able to find the pathogen's DNA. Potentially, the ear tissue might not be able to fight off an infection as well as the rest of the body, as it's kind of far away from the rest of the body, I should say. But there's a ton of potential explanations. Unfortunately, my project's not able to definitively say one reason or another. But it's a very interesting topic to think about. So
Can I ask a follow-up to that?
yes.
I thought in one of our discussions, you told me about one of the hundreds of papers you've read, something about Borrelia having evolved some mechanisms to stay stuck in the ear. Do you know anything about that? Do you remember like telling me that amazing thing?
As in, what specifically do you?
Well, I don't know. I mean, how could a bacterium like stay stuck somewhere?
Yeah, so it's an excellent question. And from what I've seen online is Borrelia has specific proteins that allow it to interact with specific tissues in a host in order to allow it to kind of go to one area or another. So even though bacteria are very simple, are a little bit more simple than eukaryotic cells, they're still able to persist and or find a way to survive in a host.
So, Sada, I see your hand up. Can you unmute yourself? Thanks.
Hello, Joe. It was a really good talk.
Yeah.
I'm really excited by your results, and I wanted to know if you have any explanation of why you got back from your Babesia positive samples, hepatozoan.
And so will you repeat that question for the,
Yes, so you're saying why my Babesia samples came back as hepatozoan?
Yes.
Yes. So what I found in the literature is that the specific portion of DNA in both of the pathogens I was looking for, I looked for the same locus. I looked at the 18S rRNA sequence. And what I found is that these two species, cross amplification is very common, meaning there's not a lot of difference between their 18S rRNA sequence. So therefore, even though my primers, specifically the hepatozoan, were really specific, Babesia may not have been specific enough. So yeah, and when I looked at the specific sequences, what I found is that the total fragment, there was a small chunk in which made the results go from primarily matching with Babesia to instead hepatozoan. So that small chunk there shares some overlap, and that's potentially why I found hepatozoan rather than Babesia. So
Thank you, and congratulations.
thank you. I appreciate it.
Question.
see someone speaking?
Yes.
Here's Nora here.
Yes.
Joe, you really did a phenomenal job at being very linear in the thought process and explaining all the pieces of all your work. So I really want to congratulate you for that.
Appreciate you.
You did talk quite a bit about the weight of the mice and the
Yes.
importance that may have or not on some of your results. And that, of course, the first thing that makes me think about is, do you look at the differences by sex, by age? Yeah.
That, as in, are you referring to if one sex is larger than the other?
Well, one sex having more pathogens than the other because one of them could be, heavier
Yes.
than the other. Yeah.
Okay, yes. When I was trying to determine the best model, when I looked at those AIC values, I took that kind of like a grain of salt, right? Unfortunately, with my sample size being 60 mice, of which only a small subset were infected with each one of the pathogens, I did not want to try to analyze the sex of the mouse because for some, like trypanosoma, all the mice that were infected were male. So there appears to be some level of significance among the sex of the mouse, but I did not want to try to explain any amount of that variation.
I'll ask a follow-up to that.
Yeah.
So I think Dr. Mateus Pinilla is asking, did you did you look to see if sex had an effect on pathogen detection? And the way you did your modeling, did you ask?
Yes.
And what did it say?
It's from what I remember, it's definitely worth looking into. And for future studies, I'm definitely going to consider the sex of the mouse. Because there appears to be some interaction and or some effect that can be explained by the sex of the mouse.
And I'll push a little more, but did sex make it into the best model that you started talking
It
about?
did not make it into the best model.
Okay.
So I considered it at the beginning, but it did not make it into what is classified as the best model, overall model.
And why do you think weight is so important?
That's a good question. Again, I can only give out potential explanations. My data did not definitively say one way or another, but from previous students, I know that, say, it could be some sort of hormone, right? Testosterone tends to be more common in males, and it lowers the overall immune system, potentially resulting in male mice having a more common infection. Or it could be that larger mice move around more, right? They tend to travel more, increasing the likelihood they come in contact with a tick. And they also could just be larger, so you can have more ticks on the mouse. So there's a bunch of potential explanations.
Could larger mice be also older mice?
Potentially, yes. That is a good point. Mice typically in the wild, they don't live very long, but that is definitely something to consider. Yes. So that's a good point.
Good. Thanks. Okay.
Thank you for the question.
Oh, there's a question in the back left.
So I know people that can't get rid of meat because they're they start sick. You know what the exact disease is called?
Yes, so it's technically a syndrome. It's alpha-gal syndrome. It is kind of complex. So instead of a pathogen causing that allergy, what it is that alpha-gal is essentially a sugar molecule that's found in red meat. So whenever you consume it, your digestive tract does not produce a reaction to it. Well, if a tick happens to bite you and pass that sugar into your blood, your body develops an immune response to it. So then whenever you consume the red meat, your body reacts, producing an allergic reaction. So it's a really kind of interesting, kind of complex syndrome, essentially. Yes. Yes, okay, sweet. It is definitely interesting. It's very, it's kind of odd. It's kind of unique. Any other questions?
Let's see. Is that everyone?
Yeah. Another?
So you've had a lot of ticks on ears,
Yes.
right? Did you take any samples from ticks themselves, or was it just strictly mouse tissue?
Strictly mouse tissue.
Okay. Would you maybe consider like looking into like what diseases ticks that are found, like maybe ticks that are found in the ears might carry, and like see if that correlates to like what you found in the ear at some point? That sounds interesting. I think
That does sound pretty interesting. I never considered that. That would be something to potentially look into.
just a thought.
Yeah.
I think those ticks are in alcohol in my bottom drawer. Not bad. That's a joke. They're they're labeled. They're in boxes. Joking. Okay. Okay. I'm looking. Oh, yes.
One more question, since you have the drawer full of creatures and ethanol, do you also have mites and fleas, which could be important vectors for a few of those pathogens?
Yes.
Lee, I'll let Joe answer regarding what he's seen.
Yes. So when we were dissecting mice, we did catch some of the fleas. The issue is the fleas were really small and they were really fast. So as soon as you humanely euthanize a mouse, those fleas take off running. So we did not catch all the fleas on the mice. We did the best we can and we caught the majority of them, but we do have them to my knowledge.
Mhm.
So it's definitely something to consider, potentially for future research.
Thank you. Congratulations. Very nice job.
Thank you. Appreciate it. Appreciate you saying that.
Okay, then I think that's it. Let's thank Joe one more time. Okay, thanks everyone for coming. A reception for Joe at my house later today if you want to come. I'm not sure, Dr. Muster, your class is dismissed or they're staying?
Yeah, that's dismissed.
Okay.
Just write a page if you haven't already and I'll upload it.
Okay, thanks
Yeah,
everyone.
thank you for showing
for
up.
coming out there. We're turning this off in a minute. Goodbye and thank you.
Great.
Okay.
You know, my science is.
All right, so let's, it's not recording.